So in the last video, we have discussed uh, N-acetylglucosamine inside endoplasmic reticulum, and in one of the earlier video we have discussed how the quality control mechanism in ER takes place with the help of calnexin and calreticulum. So after the protein that pass through this that qualify or that is approved by this quality control mechanism of ER, it will then go towards Golgi via ERIC. ERIC is endoplasmic reticulum intermediate Golgi compartment. So this transport of protein from between different organelle, that is for example between ER to Golgi, is done by COP2. ER to Golgi is done by done by COP2. And in case the protein does not form a native structure in the Golgi compartment, or it deviates from its native structure, it will again move back from Golgi to ER, and that is done by COP1. It is done by COP1. COP1 is basically COP1 and COP2 is basically vesicle protein that carries the protein from one organelle to another, and this COP1 performs from Golgi to ER, and COP2 performs from ER to Golgi. So when the protein attains its native structure, the final glycan that is attached to the protein structure when it leaves the ER has 8 mannose and 2 N-acetylglucosamine. So what happens? There are different kind of proteins that are secreted inside the Golgi. One of them is protein. There are different kind of proteins that are produced or formed inside the Golgi. Final destination of some proteins are either go outside the cell, that is intercellular proteins, and some of the proteins remains in the plasma membrane, whereas some might go to the lysosome as they might not form their native structure. So today we will discuss about the proteins that will either secreted outside the cell, or that will go in the plasma membrane. So for those kind of proteins, what kind of post-translation modification does is initially the mannose, 3 mannose are when it reaches the Golgi. In the Golgi, the first step that happens for the proteins that are secreted outside the cell or that remains in the plasma membrane is first the 3 mannose in this glycan structure is removed. We can remove any of the 3 structures. Let's just remove this 1, 2, and 3. The first step happens is removal of 3 of the mannose, and then second step that happens is addition of N-acetylglucosamine. Another N-acetylglucosamine is added. N-acetylglucosamine, and after that next step happens is 2 mannose are further removed. 2 mannose are further removed in this. So let us remove 2 mannose. 2 mannose are further removed. After that, the further step that happens is additional fucose. Fucose is added. Fucose is added, and to fucose and to N-acetylglucosamine is further added. Let's just represent fucose with the red. Fucose is added, and to N-acetylglucosamine is further added. Just add fucose here. Glucose is generally added at N-acetylglucosamine, not at the mannose. It's just general structure. It could be added in some cases. Okay. So now at this step, after that, what will happen is 3-galactose. Galactose has the same molecular weight of mannose. However, they are different in structure. So 3-galactose further. 3-galactose are added. 3-galactose are added. After that, the final step is the addition of the sialic acid. Addition of the sialic acid. 3 sialic acids are added. 3 sialic acids are added. So this kind of proteins with this kind of blackened structure will either remain in the plasma membrane, or they will secrete outside the cell. However, we need to understand one thing, that not all the N-glycosylation sites will get this kind of structure. The N-glycosylation process depends on the amount of enzyme present in the Golgi. At the same time, also on the structure of the protein, as based on the different structure of the protein, some N-glycosylation sites are more accessible to the enzyme, and similarly, if the amount of enzyme is more in the Golgi, the N-glycosylation site are more accessible by the enzyme, and vice versa is also possible. That is the reason we observe for similar protein. That is the reason we observe. That is the reason many students or researchers has observed different glycosylation on the same protein, because it either depends, because either the amount of enzyme is low in the Golgi at that time, or the protein structure is not accessible, or the N-glycosylation site is not accessible equally in every step of the secretion of protein via Golgi. Okay, so in next lecture we will discuss how the glycosylation pattern will form for the protein that the final destination is the lysosome. Okay, thank you.